All right, welcome to The Explainer. Today we are going to crack the code of life itself. We're talking about this simple 20-letter alphabet that our bodies use to, well, write the entire story of who we are.
So inside every single one of us, there's this class of supermolecules called proteins, and they are the ultimate multitaskers.
I mean, think about it. They build the fibers in our muscles that let us move, they form the antibodies that fight off infections, and so much more.
Take hemoglobin in your blood. It's just tirelessly carrying oxygen with every single breath you take. That's a protein doing its job. They are, without a doubt, the most diverse and powerful workhorses in all of biology.
So how is this possible? How can one type of molecule do so many different things? Well, the secret isn't really in the proteins themselves. It's in what they're made of, their fundamental building blocks.
All right, let's get into it. You see, all of that incredible, mind-boggling diversity in proteins, it's all built from a surprisingly simple and standardized set of components.
Yep, 20. That is the magic number. Every single protein in your body, from the collagen that holds you together to the enzymes digesting your food, every single one is built from just 20 standard amino acids. These are the only ones our DNA actually has the code for.
Okay, so what is an amino acid really? Well, at its core, they all have the same basic structure, this common backbone. You've got a central carbon atom, an amino group on one side, a carboxyl group on the other. But it's the fourth piece, the one that's different for each one, that makes all the difference.
And this is where all the action happens. That unique part, it's called the R group or the side chain. This is what gives each of those 20 amino acids its own distinct chemical personality. And that personality, it dictates exactly what role that amino acid will play in a protein.
So let's talk about these personalities. How do they actually work? Well, a really great way to start understanding them is to group them based on one simple property, basically how they feel about water.
And right away, this creates this fundamental divide. On one hand, you've got what are called nonpolar, or hydrophobic side chains. Just think of them as oily and water-hating. On the other hand, you have the polar, or hydrophilic side chains. And these guys are water-loving. And believe it or not, this super simple difference, it's one of the most powerful forces in all of biology.
So imagine a protein just floating around inside one of your cells, which is mostly water, right? All those water-hating nonpolar parts, they want to get away from the water. So what do? They tuck themselves away, clustering together deep on the inside of the protein. This is called the hydrophobic effect, and it's a huge reason why proteins fold up into their specific functional 3D shapes. But this is what's so cool. It shows how the environment is everything. Now take that exact same protein and stick it inside a fatty cell membrane. All of a sudden, those water-hating side chains are totally in their element, they're right at home. So they do the complete opposite. They flip to the outside of the protein to hang out with all the surrounding fats. Some of these amino acids are real characters, true individuals. Take proline, for instance. Its side chain is weird. It actually loops back and connects to its own backbone, which forms this rigid ring. It's a total rule breaker. It can't fit into the normal, neat structures like an alpha helix. So instead, it acts like a structural kink, forcing these sharp bends and turns into the protein chain.
And then you've got cysteine. This one has a legitimate superpower. If two cysteine molecules get close enough, their side chains can link up and form an incredibly strong bond. It's called a disulfide bond. You can think of these like molecular staples, just locking a protein's folded shape firmly in place. This is super important for proteins that have to survive outside the safety of our cells.
Okay, so here's the bottom line. The specific sequence of these amino acid letters, it is everything. And I mean everything. Changing just one of them can have absolutely massive, real-world consequences.
So what happens if you swap a water-loving amino acid for a water-hating one? Let's look at one of the most famous examples of this in genetics, sickle cell anemia. The entire disease comes down to one single tiny swap.
Okay, check this out. In normal hemoglobin, the amino acid at position number 6 is glutamate. It's polar, it's water-loving, and it helps keep all the hemoglobin molecules from sticking to each other. But in sickle cell anemia, that one amino acid gets swapped out for valine, which is nonpolar, water-hating. That one tiny change creates this sticky patch on the outside of the molecule. So what happens? The hemoglobin molecules start clumping together, which warps the whole red blood cell into that classic and damaging sickle shape, all from one letter change.
But what if we could swap these letters on purpose? See, this deep understanding of their personalities isn't just about explaining what goes wrong in diseases. It actually lets scientists become molecular engineers. They can redesign proteins from the ground up to create better medicines. A perfect example is a long-lasting insulin called insulin glargine. The problem with standard insulin is that it works too fast. So scientists went in and did some clever engineering. They strategically replaced one amino acid and added a couple of others. And these little swaps were enough to make the whole molecule less soluble at the body's normal pH. So when you inject it, it gets absorbed much, much more slowly. It was this brilliant bit of engineering that completely changed the game for millions of people with diabetes.
So, when you take a step back and look at the whole picture, what you see is just this profound elegance. The whole system is a masterclass in building incredible, mind-boggling complexity from just a handful of simple parts.
I mean, think about it. Every single function, every structure in your body, from fighting off a cold to just thinking a thought, it's all orchestrated by proteins. And every single one of those proteins is just a unique sentence written with the same 20-letter alphabet.
And that really leaves us with a pretty fascinating thought, doesn't it? We've gone from just being able to read this molecular alphabet to now being able to actively write with it. Because we understand the personality of each and every letter, we can start designing new proteins, new therapies, basically from scratch. It's opening up a whole new chapter for medicine and biology. And it really makes you wonder, what stories are we going to write next?